Hello friends, good day and it's beautiful having you back here. And today we're going to discuss bright field microscopy. Bright field microscopy, and this is one of the techniques, or you might even call it the simplest technique in microbiology, life sciences and biological sciences. I want to take us through some series of very simple techniques from there to more complex ones that we do in the laboratory, to be able to help us in our investigation. Now, today we're talking bright field microscopy. And what's bright field microscopy? This is a form or a type of microscopy where we make use of the microscope and we make use of a large light source. Sometimes we use LED light or halogen source of light, to view an object. And usually we hit the object with the light from below and then we view from above. To simply understand this, you think about your simple light microscope. Simple light microscope is making use of the bright field microscopy. You have a situation where you have your object mounted on a stage, and then you have your light source, and then your light source is hitting the object from below. And then from above, you as a scientist, you are viewing through an eyepiece and then through series of objective lenses to create an image. Now, it's important to note that for this to happen very well, many of the specimens that we deal with, they have to be stained. Sometimes many of the specimens we deal with is difficult for white light to penetrate through them, and sometimes some of them are even transparent so that when white light goes through them, they don't create an image that is useful for viewing. So many a times, many of these specimens, many of these samples have to be stained. Now there are different kinds of stains in science, Gemsa stain, Wright stain, acid fast stain, gram staining. There are many different types of stains. And the idea behind staining these specimens is just simply to create a contrast, to help our specimens to be able to absorb and scatter light. Remember that the specimen is an object, and you're trying to create an image from that object. You're trying to create an image that is clear enough for you to study, that is clear enough for you to understand. So it's important to note that this method works because of the ability of many parts within the specimen to both absorb light and scatter light. Both absorb light and scatter light. So as it's absorbing and scattering light, different components of our specimen, as they are absorbing and scattering light at different paces and abilities, it creates an image. And this image, when we view it, we're able to understand the content or constituents of our specimens. And this is quite important for us to know. Now, like I said before, I said you have a light source. So the light source could be LED light, it could be halogen light. You also have a condenser. So the light is sent through the condenser. And the work of the condenser, which you have in many light microscopes, is to focus the light, focus the source of light, let it hit that specimen very well so that the light doesn't scatter before hitting the specimen. So you have your light source, you have your condenser, then you have a series of different lenses, objective lenses. So you can have your times 4, you can have your times 10, you can have your times 100, you can have your oil immersion. These are different types of objective lenses. So these different types of objective lenses will magnify the image to different levels. You have different capacities, different magnification. And so with that too, apart from the objective lenses, you also have the eyepiece lens, which also that's where the scientist puts his eye in and is able to view many of all these specimens. So this is very important to use. Where do we make use of this bright field microscopy? In many places. For example, we use it to view bacterial samples. So if you culture bacteria from many samples, from soil, from water, from feces, from breast milk, from whatever sample it is that you're taking, when you culture this bacteria, then many a times we need to stain the bacteria depending on the kind of bacteria it is. So we could do gram staining, we could do acid fast staining, whatever type of bacteria it is. So when we stain this bacteria, then we mount this bacteria on a stage and then we use bright field microscope. It's also important to note that it's being called bright field microscopy because when you look into the microscope, you notice that there seems to be a bright white light all around, and then you have your sample that you are viewing in the middle or scattered within that bright light. That's where it gets the name bright field microscopy, because you have bright white light into which you are looking, and then you are trying to make sense of what you can see in the specimen there. So this is important. Different types of bacteria can be seen: E. coli, Staphylococcus, Streptococcus, um, many different types of bacteria can be seen in this specimen. Now, apart from that, we also use it to examine blood smears. Take samples of blood and you make a smear. You stain many times. You use either right stain or ginger stain. So if you stain these blood samples, you can view them in the microscope. It's very good to help to identify white blood cells, identify red blood cells, identify the presence of particular parasites. Especially the simplest one we do, especially in Africa, is the test for when we are trying to do microscopy for plasmodium, because we deal with malaria a lot. So different types of plasmodium, vivax, ovale, malaria. So we can see them a lot in those blood samples, and this is important for us to know. Must understand that. Also, when we do histological sectioning, so when we take thin slices of tissues and mount them in the microscope, it can help us to. We can also view, with bright field microscopy, and when we stain those our samples, we can view them and see what it is that we can learn from them. So the advantages is that it is easy to operate. It's not that expensive to get microscopes that use white light or light microscope. They are not that expensive to see. Also, the stained specimens are excellent for results. It has some few limitations too. Some of the limitations is that you can't view viruses with it, and then sometimes the contrast is not good enough. You're not able to view very closely into the cell and see what it is that you want to see. Thank you for listening. Have a beautiful day.